Alright, for this microbyte, we'll be talking about operons. Operons are coordinated sets of genes that can be regulated as a single unit. These are genes that are under the control of a single promoter, and that means they can be transcribed as a group. Operons also contain binding sites for regulatory proteins that allow the cell to promote or inhibit transcription based upon the substances in the environment surrounding that cell. We find operons in those tiny creatures that don't have nuclei, the bacteria and archaea. Eukaryotic organisms do not have operons, which is a shame. There are two types of operons, those that are inducible and those that are repressible. Inducible operons are turned on when certain molecules become available to the cell. Repressible operons are turned off when certain molecules build up in excess in the environment surrounding a cell. The classical example of an inducible operon is the Lac Operon. Most of the time the Lac Operon is off. And in the off position, the repressor protein is bound to the operator of the operon. This essentially locks the operon in the off position as RNA polymerase is prohibited from binding to the promoter and initiating transcription. However, when a bacterium possessing the Lac Operon finds itself in the presence of lactose, a series of events leads to switching the Lac Operon to the on position. This series of events is triggered when the inducer molecule lactose binds to the repressor protein, and that moves the repressor protein out of the way and inactivates it, which means the repressor protein is removed from the operator region, and with the repressor protein out of the way, RNA polymerase can bind to the promoter and initiate transcription of the genes in the operon. So those genes will then be translated, and the proteins produced serve as enzymes to help break down and digest lactose. Eventually, lactose will be depleted, and that means the cell will no longer need to make the enzymes necessary to tear lactose apart. So the whole process sort of reverses itself. As the repressor protein is freed up, it will bind to the operator once again, and thus the Lac Operon is turned off. On the other side of the fence, we find the Arg Operon, which is a repressible operon. The Arg Operon is typically in the on position because the cell needs arginine. Arginine is an amino acid, and cells are constantly building proteins. When the Arg Operon is in the on position, RNA polymerase can freely transcribe the set of genes that are ultimately translated into enzymes involved in synthesizing arginine. When levels of arginine are low, the repressor protein is in the wrong shape and cannot bind to the operator region. As the concentration of arginine begins to accumulate, arginine will bind to the repressor protein, which triggers a conformational change as the repressor adopts the correct shape to bind to the operator region. This halts transcription and switches the operon off. When the levels of arginine begin to drop, the process will reverse itself and the operon turns on once again. Excellent. Here's a GIF of the Lac Operon created by those wonderful ladies, the Amoeba Sisters. Awesome. I just love these GIFs. They're so awesome. Well, folks, that's it. Here's my dog, Callie. And as you leave, always remember, micro is magical.